So we got a good handle on meiosis. There's two stages, and we recall that meiosis 1 separates homologous chromosomes, meiosis 2 separates the sister chromatids. So now that you've looked at the video and maybe you looked it up in the lecture, you have a good understanding of meiosis. And one of the main functions of meiosis is not only to recombine chromosomes into offspring, but also to keep the chromosome numbers equal from generation to generation. So if you were to inherit all the chromosomes from your mom, all 46 of them, well, you'd have 92 chromosomes since you got both sets from your mom and dad. And they would build up, of course, extremely quickly. In some cases, in agriculture, for example, it's called polyploidy, whereby that kind of thing is induced. So some of our wheats, for example, some rye, different kinds of grains are polyploids. So they're not diploids as we are. We have two sets of chromosomes, homologous chromosomes, but some organisms have more. They might have 4 sets or even 8. I think strawberries are octoploids, so they have 8 sets of chromosomes. Anyway, if you wanted to review meiosis, I'll just go, I'm not going over every stage, but you can do that. It's in the lecture. But I would like to talk about meiosis in the context of spermatogenesis. That is the production of sperm. The germ cells are called spermatogonia. They are diploid, 2N, so they have all the chromosomes, both sets. Then they go through mitosis, making copies only. Of the 2 daughter cells, one is called a type A spermatogonium, and that just simply remains in the epithelial layer of the seminiferous tubule and just keeps dividing for the rest of its life. The other one, the type B spermatogonium, still diploid, will carry on and go through the blood testis barrier. And I'll show what that looks like in a moment. And it will go through, it's called a primary spermatocyte. It will go through meiosis 1. And after meiosis 1, the resulting daughter cells are called secondary spermatocytes. Then they go through meiosis 2 and they become spermatids or spermatids. Yeah, and this takes place from the outside of the seminiferous tubule toward the lumen, which is the inside of the tube. So they move inward. A blood testis barrier, I'll show you in a moment how that's formed. Yeah. So that's spermatogenesis, and it's not to be confused with spermiogenesis. So that's a different process altogether. That is where the spermatids are matured or converted into spermatozoa. So that's with an addition of a tail. They lose some cytoplasm, become nice and light. And we'll look at spermiogenesis in a moment. So in the blood testis barrier, this is a really great diagram. It's not too bad though. It's upside down from what we just looked at. So here are the spermatogonia. A, B. Well, they will go through spermatogenesis, and these large cells here, so this entire thing is one cell, and beside it is another cell. These are the sustentacular cells. They're very large. And the sperm that is growing, the spermatocytes are quite small. As you can see, they're quite small. And they're basically surrounded by the sustentacular cell, which has formed this junction. So this tight junction disintegrates and reforms after the spermatocytes weave their way through as they're developing. Let me write that down in words. In the blood testis barrier, some tight junctions between the sustentacular cells disintegrate and reform, allowing these spermatocytes through and isolating from blood, because they must be isolated from the immune system. Eventually, after having undergone meiosis II, the spermatids are released and washed down the seminiferous tubule by fluid from the sustentacular cells. How long does it all take? About 72 or 74 days. So it takes about 74 days for the initial spermatogonium to become a mature spermatozoon. Spermatozoon is singular, spermatozoa is plural. So a young man produces about 300,000 sperm per minute. That is about 400 million per day. And then they last, if not ejaculated, they last about 60 days that they're viable, but then they are reabsorbed into the body. So spermiogenesis is the maturation of the sperm. So here's a spermatid. There's the nucleus. The nucleus is the most important thing to get to the egg because that contains the chromosomes. Um, there are these little vesicles called acrosomal vesicles. No tail yet. So there's a basal body, which consists of some microtubules, and it extends microtubules out to form the flagellum. Excess cytoplasm is released. In order that this spermatozoa is light and maneuverable and can make its way through the entire system of ducts of the male, the vagina of the female, and then into the uterus and the uterine tube. So that's the transformation. This is the acrosome up here that forms containing acrosomal enzymes. So the spermatozoa is a little pear-shaped at the front end, about 4 to 5 microns long, very, very small, about 500 would fit onto the head of a pin. Contains the nucleus, an acrosome, and the basal body of the tail of the flagellum. And the nucleus contains a haploid set of chromosomes, of course. The acrosome contains enzymes. The enzymes are required to break into the egg. So we'll talk about that in development and fertilization, where it takes many sperm to actually get through the outer coating of an egg, although only one sperm will be introduced into the egg in the end. And then there's this really fast block where no other sperm can get in. Now, the tail has 3 regions, a midpiece that contains the mitochondria. The mitochondria are large because the sperm requires a lot of energy, and the vagina is an anoxic kind of environment, so low in oxygen. So it's around an axoneme of the flagella and the mitochondria produce ATP. The axoneme, which is the center part of the flagellum, surrounded by fibers. The end piece is just a very narrow tip of the flagella. So let's look at what that looks like. There's a spermatozoan. The acrosome with acrosomal enzymes that will be very instrumental in the end of getting into the egg. The nucleus, look at how large the nucleus is compared to the rest of the cell. Now the nucleus pretty much takes up the rest of the cell. The basal body, it provides the initial movement of the flagellum. So the microtubules of the flagellum are attached to the basal body. And the end piece is very small. The spermatozoum. So the basal body is there to begin the movement of the flagellum. So the flagellum is a combination of microtubules that are positioned or structured in, there's 2 in the middle and 9 pairs around the outside of the flagellum. And it's fairly complex, but not too bad. So there's proteins that they're called dyneins, and they bind and release and bind and release over and over, which produces the movement of the flagellum. And that is where I'd like to end. Thank you for watching.